In this lecture, we're going to focus on the physiological consequence of generating an action potential in the axon. And it's a beneficial consequence because this is the mechanism of how motor neurons activate skeletal muscle fibers. But there are factors that do affect the speed at which that action potential can travel along a neuron. The first aspect is the axon diameter. Larger diameter axons, so not all neurons are the same size, can conduct those action potentials faster. And the reason is that the larger diameter of the axon of that neuron provides more room for movement of the charged particles. And very often these larger diameter axons possess a greater density of ion channels. So the ion channels are proteins embedded in the membrane, and per surface area of membrane, there are some neurons that have more of these proteins embedded. And so it refers to the channel number of channels per surface area. And the reason that's an advantage is that because if the next channel to open up, the next protein to open up to allow sodium ions to flow in is closer to the one that just opened, that next one is going to open up even faster, which then will open up the next one faster. So it doesn't require the time for the electrical signal to slowly reach the next channel. If the next channel is closer, it's going to discharge sooner and move the entire propagating action potential signal down the axon faster. The degree of myelination is critical here because of this myelin, which is made up of phospholipids, provides that electrical insulation and prevents the leakage. Once again here, preventing leakage means that the ions that are responsible for the different phases of the action potential are confined closer to the axon membrane. And the physiological state will also affect the speed at which that action potential can propagate. And this is particularly pertinent for muscle cells that we'll cover in another unit. But in order to generate the next action potential, because these action potentials occur in sequential as sequential events, ions have to be redistributed. And the sodium potassium ATPase is the so-called sodium potassium pump, which is an enzyme embedded in the membranes of the neurons and muscle cells. And so this is an enzyme that is an active enzyme, which means that we have to expend metabolic energy in the form of ATP for this enzyme to redistribute those ions back to where those ions belong. And so muscle fatigue is referring to a reduction in muscle force generating capacity. And in that case, we see these very large energetic changes inside the muscle cell. And if that occurs, the ability to operate the pumps will decrease. If that decreases, the ability to restore the ions back to their resting concentrations will take longer. It'll take longer to propagate an action potential along the membrane of those cells. On this slide, we're going to take a look at the sequential steps, and when we get to the muscle physiology unit, we will revisit this. But these are the overall steps that take place that translates a neuron action potential into activation of a skeletal muscle cell. And so we've already talked about the transmission and the propagation of an action potential along a neuron, started up in the cell body, in the spinal cord, and it continues all the way along the axon to where it connects and plugs into a muscle cell. And the end of that axon, the terminal axon, is where the electrical signal has nowhere else to go, but it will trigger another event. That other event is the opening of voltage-gated calcium channels. So we have proteins that are specific to the movement of calcium that will open when triggered by an electrical event. And that electrical event is that action potential traveling down distally along a neuron. So these are calcium channels that are embedded in the membrane of the terminal axon, not the skeletal muscle cell. And when these calcium channels open up, that will eventually trigger the release of the neurotransmitter acetylcholine because calcium has entered the cell. And we'll see that graphically on the next slide. Once the terminal axon releases its neurotransmitter, acetylcholine, acetylcholine will move across what's called a cleft. That is the very small space between the terminal axon and the muscle fiber and bind to a class of protein receptors called nicotinic receptors on the specific region of the membrane, muscle fiber membrane, called the motor end plate. And that acetylcholine will trigger the opening of ligand-gated sodium channels. So these are sodium channels. They will allow sodium to flow into the muscle cell. But what opens these sodium channels? These are ligand-gated. So it's going to take a chemical to do that. That chemical is acetylcholine. And so once those sodium channels have opened up, there's going to be a net movement of the positive charges sodium into the cell. Remember, the cell is already polarized at -60, and because we introduce all those sodium ions, the cell will become depolarized and produce a localized action potential at that specific region of the membrane of the muscle cell near the axon, and we call that the end plate potential. And the end plate potential will then generate a subsequent action potential that will propagate and move along the entirety of the membrane of a skeletal muscle cell. And that spreading of that action potential will eventually cause muscle contraction that we'll explore in greater detail in the muscle physiology unit. So we use this graphic here to illustrate these different steps. So the yellow here represents the terminal axon. This is in the actual muscle, and this is one muscle cell that we see here in the red. The blue represents the skeletal muscle cell membrane, and the blue represents the membrane of the motor neuron. This isolated region of the muscle cell membrane, it is continuous with the entirety of the skeletal muscle cell, but if we just look at this portion in close proximity to the axon, this region is called the motor end plate of the muscle fiber. Now, what we have in the yellow is, remember, we have that action potential, that electrical signal traveling distally along the axon to where the axon plugs into the muscle cell. Now, what is a unique feature of the terminal axon is that it has a wider shape. It is expanded. It's larger at the very terminal end because it has to have room for what are called synaptic vesicles. And these synaptic vesicles are these small carriers of neurotransmitter called acetylcholine. Now we'll jump over to the larger expanded view here. And so we have our number 1 at the top left-hand corner of this boxed image, and that's our action potential that's traveling. Now the action potential, when that electrical signal reaches the terminal part, it will cause these calcium channels to open up. And these are proteins embedded in the membrane that are sensitive to changes in the voltage. What's causing the change in voltage? It's the traveling action potential. Now recall, the calcium concentration in the extracellular fluid is considerably higher, much, much higher than that in the cytosol. So as soon as these proteins change shape and open up, calcium will flow down its concentration gradient and rush into the terminal portion of the axon, as illustrated in number 2. Now that calcium will trigger the vesicles to fuse, F-U-S-E, fuse with the interior of the membrane of the terminal axon. And because these vesicles are surrounded by phospholipids, they form chemical bonds very easily with the interior of the terminal axon, which causes these vesicles to open up and to release acetylcholine, the neurotransmitter, which diffuses across this very small gap. This is called the synaptic cleft. Okay, so the synaptic cleft is the very small gap between the terminal axon and the muscle fiber membrane. And the acetylcholine will find its way onto two different proteins. The important one here is that it binds to these a particular class of receptor called the nicotinic cholinergic receptor. And this receptor functions as a sodium channel. And that sodium channel will open up and sodium will rush into the cell. This diagram is a little bit misleading in that it suggests potassium moves this way as well. Potassium moves through a different channel. Potassium has its own channel for it to move out. But this number 4 here captures the fact that the neurotransmitter will trigger an action potential isolated at this particular region. This was ligand gated, but because we have this new electrical event, this electrical event will set up a voltage triggered action potential for the rest of the membrane, and that's what generates an electric current along the membrane of the entire muscle cell. And that's what we see here illustrated in number 6. The action potential will continue to travel, and after this event, we no longer need a neurotransmitter for this current and this action potential to travel. It's just a change in the voltage that took place that will proceed to the next one, and to the next part, and to the next part. The last part here, we see that acetylcholine is eventually degraded by acetylcholine esterase. And it's an enzyme that will take acetylcholine, degrade it, and will recycle part of it, the choline part of it, will recycle it back into the neuron, where it will eventually be metabolized and repackaged into new neurotransmitter. And the function of the acetylcholine esterase is to stop the activation of the muscle cell. And that's functional because that provides a degree of regulation so that muscle fibers do not contract continuously in an uncontrollable way. It's similar to the brakes on a car. The brakes on a car are designed to regulate the movement of the car, the speed of the car, by stopping the movement and by slowing down the movement. That's what acetylcholine esterase does here. It prevents the overcontraction of a muscle cell. So this ends our final lecture here on part 3 of our discussion.